Okay, hello everyone. Thank you for joining the Division of Rheumatology Grand Rounds. A couple of reminders before we start the session. This session is currently being recorded and will be posted later on our website. There will be a Q&A session at the end, so please add your questions to the Q&A feature and not in the chat box. And now, so it's easier to moderate. And now I'll let Dr. Lude introduce our speaker for today.
Thank you, Danielle, and good morning, everyone. It is my privilege and pleasure to introduce today's speaker, Professor Christian Frezza. Dr. Frezza received his PhD training at the University of Padova, Italy, studying mitochondria in apoptosis. He subsequently went to the UK and did his postdoctoral training on metabolomics with a focus on tumor biology. And he's currently Alexander von Humboldt's Professor of Metabolomics in Aging at the University of Cologne in Germany, where he's leading a large group of students and staff. Dr. Frezza is extremely well published with more than 250 publications, many of which are highlighted in prominent journals such as Nature, Cell, and EMBO. And today he will share with us some of his recent findings on mitochondria and metabolism in regulation of cancer and inflammation. So Dr. Frezza, welcome, and please take it away. And I think we need to change the display settings again.
What do you see?
I see two screens.
Two screens? Okay. We try. Okay, is it okay now? Oh, wait. Okay, I share it again. Okay. Do you see everything correctly? The pointers and
Perfect.
Okay,
perfect.
Okay. Thank you very much, Christian. It's great to be here, and thanks a lot for the invitation. Today, I want to give an overview of the work that we are doing in our laboratory, which is focused on understanding the role of fumarate hydratase in cancer. And at the end, I will also show some recent findings that were made that connect, surprisingly, fumarate hydratase and mitochondria with inflammation. So let me start from the overarching framework of our laboratory. As you know, during the process of cellular transformation, cancer cells undergo profound metabolic changes. These have been charted over the last 20 years, let's say, with the advent of metabolomics. And we now know that these metabolic changes are required for biosynthetic purposes. They are needed to generate amino acids, proteins, macromolecules to allow for the cancer cells to proliferate. As initially postulated, metabolic reprogramming during cancerous transformation is also needed to fulfill the energy requirements of the cancer cells. And more recently, it has been shown that this metabolic reprogramming is really essential to provide cells with antioxidant molecules. These molecules are needed during early phases of transformation, but more importantly, during extravasation of the metastatic cancer cells, because they experience a strong oxidative stress, and therefore, via these antioxidant molecules, the cancer cells can survive. However, this is not what we are interested in, at least not our main focus. Actually, we are interested in almost opposite perspective. That is, we are really keen on understanding whether this metabolic dysregulation can at all influence the process of cellular transformation. And why are we interested in these important questions? Well, about 20 years ago, or now I would say 25 years ago, some seminal papers were made that connected mutations of metabolic enzyme, such as succinate dehydrogenase and fumarate hydratase, and later of isocytate dehydrogenase, to transformation into hereditary and sporadic forms of cancers. And these were groundbreaking discoveries because for the first time, they connected mutations of core metabolic enzyme to cancer. And this really puzzled the entire field. So our laboratory is really focused on understanding how and why mutations of fumarate hydratase drive these hereditary forms of cancers. And I want to give you a brief of an overview of fumarate hydratase and this hereditary cancer syndrome to which its mutations are linked. So first of all, this scheme will come up often during the talk. So to familiarize you with this is a big mitochondrion, and this is the Krebs cycle or the 3-carboxylic acid cycle. As you know, without going into many details, but the Krebs cycle is a vital uh, node of metabolism which converts nutrients that come from glucose, glutamine, or fatty acids and amino acids into metabolites and reducing equivalents that are needed eventually by oxidative phosphorylation to generate ATP. Fumarate hydratase is the enzyme that converts fumarate into malate. This is very important for the following part of the talk. And it's important just also to see a little bit the chemistry behind that because it matters, as you will see. So what was shown in 2002 in a seminar Nature Genetics paper was that fumarate hydratase mutations are the predisposing factor of this hereditary cancer syndrome called hereditary leiomyomatosis and renal cell carcinoma, also known as HLICC. And what was found at the time was that patients inherit usually one mutant copy of fumarate hydratase, and during the course of life, due to a process known as loss of heterozygosity, fumarate wild-type allele is lost, and this gives rise to a unique set of tumors, benign tumors of the skin and uterus. These are called fibroids. These are technically benign in that they do not metastasize and they do not kill the patient, but they are extremely painful and they need to be surgically removed quite routinely. And in a proportion of the patients, fumarate hydratase loss give rise to this aggressive form of renal cell carcinoma called now fumarate hydratase deficient renal cell carcinoma. This is a unique entity within the kidney tumors, and it's the most aggressive tumor type, characterized by significant metastatic potential. So why then mutations of fumarate hydratase, a well-established, almost an housekeeping gene in the mitochondria, are associated with cancer? And when I started to address this question, I actually came from a purely mitochondrial bioenergetic field. And the first thing that came to my mind was how is it possible that a cancer cell survives without a key functional Krebs cycle, which we know actually until those years, until the early 2000, it was believed that any mutations within the Krebs cycle would be lethal. So here, this is what led us to investigate finally the metabolic changes or metabolic adaptations in fumarate hydratase deficient cells that I just want to tell you about. As you will see, this hope will be important also from a clinical point of view. So the question that I want to address now is how cells survive without fumarate hydratase loss. And the underlying hypothesis that we laid at the time was that cells must undergo some sort of metabolic adaptations in order to compensate for the loss of fumarate. So the first thing that I did, this was back in my postdoc time, and eventually more recently another PhD student helped in this endeavor, we generated cellular models of epithelial kidney cells, either from the mouse or from humans, to investigate fumarate hydratase loss. In the mouse model, we capitalize on an inducible fumarate hydratase efficient mouse, and we took the epithelial kidney cells, immortalized them, and then we obtained two fumarate hydratase knockout cells. Without going into details, you can see these are the mouse cells that will probably show most of the time in the last part, the next part of the talk. And you will see that we also generated reconstituted fumarate hydratase cell lines just to assess dependency on fumarate hydratase on the phenotypes that we observed. In the case of humans, we also use these epithelial kidney cells. These are commercially available HK2 cells. And with CRISPR-Cas9, we generated different clones of fumarate hydratase deficient cells. So first thing that you will already notice here is that we managed to create fumarate hydratase deficient cells. This implied that somehow these cells manage to undergo some metabolic adaptation to compensate for fumarate hydratase loss. And we started to charter these metabolic adaptations quite extensively. And I will give you a very brief overview here. So the first thing that we observe, which is now quite established in the field, is that whenever a mitochondrial defect occurs, the first thing that one observes, along with defect in hardcore mitochondrial function, is the upregulation of glycolysis. How did we do and how did we measure so? Just to give you an example, here this is what is called a seahorse assay. The seahorse is the instrument that allows us to measure at the same time oxygen consumption rate and extracellular acidification rate. That is the surrogate for mitochondrial function and the glycolytic rate, respectively. So if we go a little bit deeper here, what we find is that the wild type cells here, and these are the two knockout clones, we see that in the fumarate hydratase deficient cells, there is a significant increase in extracellular acidification rate. That is, there is increased glycolysis, and this is paralleled by the expected defect in oxygen consumption rate. That is mitochondrial function. So this is pretty much standard. It was expected back then, but it was good to see that cells actually managed to arrange this defect in mitochondria by upregulating glycolysis. And now this is very well established in cancer and many other conditions that I'm sure you are familiar with. The thing that was not that clear at the time and was that in the absence of glucose entry into the mitochondria, we didn't know actually how the Krebs cycle will be filled. And we realized at the time this was becoming another important carbon source for the mitochondria, that cells and the mitochondria eventually depend on glutamine for the carbon fluxes. So what we found was that the cells, fumarate hydratase deficient cells specifically, take up significant amount of glutamine that fuels this linearized version of the Krebs cycle and eventually accumulate fumarate. And we found at the same time something quite intriguing, that imagine this is now a linearized version of the Krebs cycle. So it's a linear pathway that is blocked by fumarate hydratase loss here. So we found that the cells also engage into what we called a carbon escape valve. In order to avoid saturation of the pathway, some of the carbons are shunted out of the mitochondria and they engage into a pathway known as the heme biosynthesis and degradation pathway that is increased in fumarate hydratase deficient cells. We could see that indeed the fumarate hydratase knockout cells increase the production of bilirubin that is released in the media as the end product of this entire pathway. So now you have seen that upon fumarate hydratase loss, the mitochondria undergo this metabolic switch, and they switch from glycolysis to glutaminolysis. This is how it's called. And again, also this one is a pretty much established change in the setup in cells with defect in mitochondria. But the main metabolic hallmark of these cells is the striking accumulation of fumarate. Fumarate is accumulated in both the mouse and the human cells to high millimolar levels in the cells. So fumarate now is likely the most abundant metabolite in the cells, and it reaches levels that only ATP, for instance, and glutathione can reach. And this has, of course, very important implications for the cells. So fumarate at this level will affect a lot of reactions that normally would produce fumarate. And I want to, again, just to give you a snapshot of what we found. As you might remember from biochemistry, the urea cycle releases fumarate in a reaction catalyzed by argininosaxonate lyase. And just again, for those of you who not remember or not familiar, the urea cycle is another metabolic pathway coincidentally discovered by Krebs, who also discovered the Krebs cycle. And it is used for amino acid degradation. And there's a series of reactions that eventually lead to the formation of argininosaxonate, and the reaction catalyzed by argininosaxonate lyase releases fumarate, from adenylosuccinate and arginine is produced. However, when fumarate is accumulated to these very high levels, the balance of this reaction catalyzed by argininosuccinate lyase is shifted toward the substrate, in this case, argininosuccinate. And now what we found is the cells take up large amounts of arginine and use it to call buffer fumarate, and they produce a large amount of argininosuccinate. And as you can see, argininosuccinate now is produced to very high levels in these cells. Something similar happens to another biochemical pathway known as the purine nucleotide cycle. In this case, the reaction is catalyzed by adenylosuccinate lyase, and it releases fumarate from adenylosuccinate. And this reaction is important to regenerate AMP when AMP starts to accumulate. And what we found is also adenylosuccinate is strikingly accumulated in fumarate hydratase deficient cells. And this was also recently reported by the lab of Heather Christoph in California and Los Angeles. So along with these very dramatic biochemical changes, there is another very important reaction that fumarate can carry out. And this is now related to the chemistry rather than to the biochemistry of fumarate. This is why I've asked you initially to keep in mind the structure of fumarate. As some of you might recognize, fumarate is an alpha-beta unsaturated dicarboxylic acid. This makes this carbon alpha and beta mild electrophiles and subject to nucleophilic attack from reactive thioresidues. And this can be residues of cysteine, and this leads to the production of a molecule called 2-succinylcysteine, so a free adduct between cysteine and fumarate. And this reaction is called succination. And succination can also occur against the cysteine residue of glutathione. And of course, you can imagine here we just showed the measurement of succinate glutathione. This is another molecule that we and others identified. And importantly, this will also affect the availability of glutathione and therefore will affect the redox balance of the cells. But I don't want to go into details about that. So what I told you now is, and this is just a summary, is that upon fumarate hydratase loss, there are profound metabolic changes. We can distinguish these changes into two sides. The first are compensatory metabolic adaptations that are needed to compensate for the mitochondrial defect that arises. So this change from glucose to glutamine, and this changes in mitochondrial activity. But then there are changes that we call fumarate buffering systems that are needed for buffer fumarate that reaches very high levels, and therefore fumarate now is bound to cysteine, to glutathione, to argininosuccinate to keep its level viable. This is at least the current hypothesis. Now, you might be wondering why I've spent this 50 minutes talking about biochemistry. Well, there are reasons why we believe these are important. First of all, these metabolic changes that I described are pretty much unique to fumarate hydratase deficient cells. So the implications are that, first of all, we can detect these changes in vivo, in mice, and also in patients. And this is very important because HLSCC is a hereditary cancer syndrome, and therefore it will be very important to be able to track the NCO of fumarate hydratase loss. And just to give you an idea, in the fumarate hydratase deficient mice, we can find fumarate, 2-succinylcysteine, argininosuccinate in the urine of these animals, but we could also find these metabolites in the tissue samples. We could find fumarate accumulated in the tumor samples from these patients, along with the succinated metabolites. But a recent study by the Zhang laboratory also showed that even in the blood of HLRCC patients, we can find succinated metabolites such as succinylcysteine. So, and they propose that this is very important because we can use this to track, for instance, not only severity of the disease, but also the success of therapy. The last thing that, unfortunately, I don't have the time to show you, but these metabolic changes are also unique from a targeting point of view. We can leverage these changes to selectively kill fumarate hydratase deficient cells, sparing the wild type, achieving what we called back then metabolic synthetic lethality. And indeed, we have shown that by targeting some of these reactions, such as heme oxygenase within the heme pathway or arginine uptake and so forth, we can selectively target these cells and we can spare the wild type cells, so increasing the therapeutic window. So now I will want to move from understanding how these cells actually survive, in the kidney at least, to understanding a little more how does the loss of fumarate hydratase cause transformation. And this is still an open question. Now, we and others have focused on fumarate because I told you the fumarate is highly accumulated in the cells. And I've shown you also that fumarate has very important biochemical and chemical functions. And indeed, if we go back to the idea of succination, you might imagine that along with cysteine and glutathione, fumarate can also bind and inactivate a plethora of cysteine-containing proteins. And here I'm presenting a Buchanan plot of the succinated peptides in the fumarate hydratase deficient cells. Of course, this is a Buchanan plot, all shifted to the right. But what we can see here are all the peptides or proteins that we found to be succinated. Some of these are actually already known. And for instance, GAPDH has been already found to be succinated. We have others that were currently investigated. But the point here is that succination can lead to different effects. And at the moment, we do not know whether all of these are inactivating functions or the function or activating the function. So if you're interested in succination, Eva Guberovic and I in the lab have recently published a review on this. And here we charted all the succinated targets in the cells reported so far, for which also there has been an investigation of the function. And you can see that here. What I want to point your attention is a few aspects of this, because this could be important also for your research, I believe. We have recently generated an antibody that is now commercially available that recognizes succinated proteins. This is how it looks. For instance, in the wild type and knockout human cells, these are all the succinated proteins that you can find. And this could be useful as a diagnostic tool. For instance, if you want to do immunofluorescence staining, we can see succinated proteins in fumarate hydratase deficient cells. But just to give you an idea, this is now being proposed to actually accompany pathology studies on the tissue, both in the kidney and the uterus of HLICC patients. Just to give an idea, the staining is genuinely black and white between the normal tissue and the affected tumor tissue. From a functional point of view, I also want to present one example of a protein that is succinated that has very important consequences for the cells. So one of them that was characterized already, a few years ago now, more than a decade, is KIAP1. You might know KIAP1 because it is the well-established negative regulator of a transcription factor called NRF2. When KIAP1 is bound to NRF2, NRF2 is allowed to be degraded in the cytosol through proteasome. However, KIAP1 is a thiol-rich protein that contains thiol residues that can be succinated by fumarate. And indeed, it was found that fumarate can bind to cysteine residues of KIAP1. This disrupts the binding to NRF2 and that eventually allows NRF2 to go in the nucleus and activate a powerful antioxidant response. This might ring a bell. I told you at the very beginning that antioxidant response is actually very important for cancer. And indeed, it's been proposed, even though not formally demonstrated, that NRF2 activation is vital for the transformation of fumarate hydratase deficient cancer. What is important, I just want to give you another, I find very intriguing hint, is that this antioxidant response lead to the expression of hemoxygenase or HO1, which is the rate limiting step for this heme biosynthesis that I told you at the beginning is vital for the cells to survive. And also within the antioxidant response, we have genes that allow for increased glutathione biosynthesis, which I also told at the beginning that it's constantly removed by this reaction called succination. So these set of reactions that are led by NRF2 expression are actually helping to allow the cells to survive and also to buffer this increase in fumarate. So beside this chemical activity of fumarate in the cells, fumarate has also a very important biochemical consequence in the cell. You might know this class of enzyme called alpha-ketoglutarate dependent dioxygenases. And as you will see, fumarate can control this reaction. Just in brief, alpha-ketoglutarate dependent dioxygenases are enzymes that hydroxylate either protein residues or other substrates, as you will see in a second. These reactions require alpha-ketoglutarate and also oxygen. These are oxygen-dependent enzymes, and they produce succinated carbon dioxide. One of the most established example of these enzymes are prolyl hydroxylases, which are known to hydroxylate HIF, and HIF hydroxylation is also known to lead to HIF proteasomal degradation. So this is a very elegant mechanism that controls HIF stability. HIF is an epoxy-inducible factor, depending on oxygen tension. So when oxygen is low, this reaction is blocked. HIF is no longer hydroxylated, and it allows its transcription of activation of the epoxy response. What it was found is that fumarate, which has a similar structure to alpha-ketoglutarate, when it is produced at very high levels in the cell, it can block this reaction, leading to an aberrant stabilization of HIF even when oxygen is present. This is a very strange configuration, which is called pseudo-hypoxia, whereby HIF is present along with its transcriptional response, even when oxygen is present. And it has been proposed that this pseudo-hypoxia is very critical for the transformation and tumorigenesis of HLA-CC. Just to give you an idea how this works, we found that fumarate blocks the catalytic activity of prolyl hydroxylases, allowing HIF to be stabilized. And you can see this Western blot. These are the wild-type cells and the knockout cells in the insert. And this is under normal oxygen tension in the tissue culture. You can see that HIF here is truncately stabilized along with HIF target genes. And what is intriguing, I want also to point this out, is that within this within this HIF response, there's a series of glycolytic enzymes that are responsible for the switch towards glycolysis and the inhibition via pyruvate dehydrogenase kinase and inhibition directly of pyruvate dehydrogenase of glucose entry into the mitochondria. So again, another transcriptional regulation of the biochemical changes that I described at the beginning. And finally, another set of reactions that are inhibited by fumarate, also within the realm of alpha-ketoglutarate dependent dioxygenases, are the TET enzymes. TETs are enzymes that instead of hydroxylating proteins, they hydroxylate methyl-cytosine in DNA. And as you know, this hydroxylation of methyl-cytosine leads to the subsequent demethylation of cytosine, which is known, very broadly speaking, to elicit gene expression. So fumarate, similar to what I told you before, can block this reaction. And this, of course, changes the epigenetic landscape of the cells, leading to epigenetic hypermethylation and gene silencing. What we found was that fumarate can inhibit that. And we found that one of the main output, at least this is what we show in this hypermethylation, is the inhibition of a family of antimetastatic microRNAs called microRNA 200, which are very strongly suppressed and epigenetically hypermethylated. And why this matters? Because we found that when these microRNAs are suppressed, they no longer de-repress they no longer repress this transcription factor called ZEB1 that is important, implicated in an epithelial to mesenchymal transition. This is a process that we know is associated with a transition from an epithelial phenotype to a mesenchymal phenotype, and this is usually linked with increased cell migration and metastatic potential where we're talking about tumors. So we believe that by repressing these microRNAs and the activation of this vimentin and this EMT, these cancer cells acquire an important aggressive nature. And we found that indeed fumarate hydratase deficient cells have increased migration, and they have also increased formation of spheroids that is dependent on fumarate hydratase. And just to give an idea, we also show that sole addition of fumarate in wild-type epithelial cells can give rise to a similar suppression of microRNAs by epigenetic suppression, the activation of ZEB1 as a consequence, and an epithelial to mesenchymal transition. So I give you now an overview of not only the metabolic changes, but also the transcriptional changes that fumarate can elicit. And I suppose now we have reached a point where we might all be confused, and you might be asking, okay, so this guy is telling me that these cells undergo, first of all, profound metabolic changes, then they described a series of oncogenic or putative oncogenic cascades, but we start to, again, be confused because we don't know exactly when a tumor starts to develop which of these transcriptional cascade matters. And indeed, this is a big question that we are trying now to address in the lab. When we take, for instance, into account tumor that has been there for maybe 30 years or more, all the signatures are inevitably intertwined, and it is impossible to understand and to address which of this signature is pro-tumorigenic and which is pro-survival and which is pro-metastasis. So we started to to ask the question of what is the chronology of fumarate hydratase loss. That is, what is the chain of events that undergo cells that lack fumarate hydratase? The other important question that you might also be wondering is, okay, here is Christian told me just about the loss of fumarate hydratase in the kidney cells, but what happens in all the other tissues, and in particular the tissues that are affected by the tumors, that are the smooth muscle of the skin and uterus. So really what we are trying to do, and this is work in progress, is try to decipher the chronology and tissue specificity of FH loss. And to do this, without going into details, we have generated an inducible fumarate hydratase knockout model by crossing the standard fumarate hydratase flux allele with a ROSA26 Crea recombinase. And without going into jargon, what matters is that we can inactivate fumarate hydratase by giving tamoxifen to an adult animal, and then we can look at the response in a time-dependent manner in different tissues that we harvest. The underpinning idea is that we will be able to see what is the response to FH loss in different tissues and identify whether there are tissues that are more resistant to fumarate hydratase loss and tissues that are less resistant, and maybe in this tissue the fumarate hydratase loss is lethal and would be negatively selected. Just to again give it a snapshot of our findings that still a work in progress, I give you some prototypical responses. So upon 10 days of fumarate hydratase inactivation, we found that the kidneys are pretty much unaffected by by fumarate hydratase loss. And I'm showing you here, in all these images, fumarate hydratase loss is equal across the tissue. So here, the fumarate hydratase loss doesn't seem to have overt structural changes. This is, by the way, hematoxylin neozyl staining that you might be familiar. However, we found a very striking response in the small intestine of the animals. As you can see, the villi that here are perfectly preserved are now distorted. The architecture of the villi is completely damaged. We found this accumulation of massive vacuoles that we don't know yet what they are. But this is not a response that occurs in the entire gut, and it's specific to the small intestine. Because in the colon, for instance, the effect of FH loss is not as dramatic, and we see only modest changes in the tissue architecture. So what does this mean? It means that when you look at broader in the whole body of the mouse, fumarate hydratase loss indeed gives rise to a different response in different tissue. And one of the hypotheses that we had, based also on what I told you at the beginning, is that maybe the metabolic adaptations that occur will be different in different tissues. And indeed, we have some evidence of this. Now, if we measure fumarate in choosexinocysteine, which is one of the readouts of fumarate accumulation, we found that in the kidneys, fumarate doesn't accumulate to very high levels, but choosexinocysteine accumulate very early, suggesting that indeed here choosexinocysteine might be working as a buffer. However, we found in the small intestine, fumarate accumulates to very high levels, and along with choosexinocysteine, maybe suggesting that here there is less buffering capacity, and this could explain, at least in part, the death in this tissue. And in the colon, the phenotype is in between. We have some fumarate accumulation, but 2SC is not very elevated. So this is just to give you an idea that there is indeed a tissue-specific response. But now, looking at the conserved response, we perform a transcriptional analysis of all the tissue, and this is a Volcano plot of the differentially expressed genes and the pathways at day 10 again upon tamoxifen induction, and you can see in a color code the kidneys, the small intestine, and the colon. And what surprised us was that along with some of the expected signatures, such as the hypoxia in the kidney or the hypoxia also in the small intestine, we found a huge enrichment for inflammatory genes that, to be honest, we never encountered before. And when we look at the genes that belong to the signatures, we found that across all the tissues, even at 5 days, we found a very striking upregulation of this gene called EF202B that we didn't know at all at the time. So we needed to study a little bit. This is where maybe our field started to overlap with yours. We found that EF202B is a scaffold for cytosolic DNA that elicits, usually is utilized in the pathway of cytosolic DNA sensing, which eventually converge into the expression of interferon response genes. And the two pathways that are implicated are, at least in part, AIM2 and the SIGA-STING, which I'm sure you are familiar with. What matters still is that this pathway will lead to the activation of some inflammatory genes. These are the ones that eventually shoot up in our analysis. So in order to study this in a reductionist approach, that is what we usually do to avoid also any problem with the whole body of the mouse. We took again the epithelial kidney cells of this mouse model. We can inactivate fumarate hydratase with tamoxifen, and you can see that fumarate hydratase is ablated very quickly. And then we studied the presence of cytosolic DNA. Also here, I don't have to go into details. I'm pretty sure you know what this is. This is a cell where we stain for DNA. And the majority of DNA is in the nucleus of the cells, but a proportion of DNA is located in mitochondria. And here we stain the mitochondria magenta. You see the mitochondria are this magenta spaghetti-like structures. So here is the main finding for which we are still excited about. This is just a snapshot of the mitochondria in these cells. And you can see now better the mitochondrial nucleoids very clearly punctated. Upon fumarate hydratase loss, we found two important things. The first one is that indeed we started to see cytosolic DNA. We can see it as these speckles that appear already very early on. Day 6, we started to see some, but this accrues over time. But this was also accompanied by profound changes in the mitochondrial structure. You can see that from this spaghetti-like structure, the mitochondria become swollen and enlarged. And the mitochondrial DNA is very coalesced. And we started to speculate at the time that some of this cytosolic DNA was coming from the mitochondria. Without going into all the details and cutting a long story short, we found that indeed upon fumarate hydratase loss, the mitochondria undergo profound metabolic changes of the crystal. And this is something that we are currently investigating. This is a beautiful super resolution image with PK orange, where you can see actually in the live cells the crystal, and you can see how the crystal structure changes. But this is also accompanied by a release of mitochondrial DNA. And this we found that this mitochondrial DNA eventually is responsible for the activation of SIGA-STING, at least in part, and the inflammatory signal. So you can see here just a snapshot of what we call vesicular mitochondrial DNA release. We eventually identify the mechanism, but I don't want to go into details. We can discuss it later. And this is a summary of the findings. Upon fumarate hydratase loss, very early on, we observe profound changes in mitochondrial structure and ultrastructure. And through a mechanism that we are still investigating, there is a formation of mitochondrial derived vesicles that release mitochondrial DNA in the cytosol and eventually activate the SIGA-STING pathway. So to conclude, what I showed you is that by elucidating this chronology and tissue specificity of fumarate hydratase loss, we started to pin down some important hallmarks here. First of all, we could see that very early on, we have profound metabolic changes, and we believe these are really probably one of the primary effects of fumarate hydratase loss. Thanks to opening the window of investigation, we were able also to find out that there is a convergent inflammatory response that we didn't see before, because this signature was probably obfuscated by more profound transcriptional changes driven by HIF and the antioxidant response. Now we still see, and we need to pin down where exactly and when exactly the antioxidant response rises and when all the others are occurring. But we found, remarkably, that the eventual phenotypic response is different between tissues, and we found examples of what we call permissive tissues, such as the kidney, where fumarate is maintained low, or at least to viable levels, but in the small intestine, for some reason that we are investigating, fumarate reaches very high levels, and this is associated with cell death. Whether this is the cause, we don't know, but this is just the current hypothesis. Now, to conclude, just a few more slides for you, and with some more clinical relevance. First of all, you might be wondering, okay, we have done all this work in cell lines, and yes, okay, is this relevant to patients? So unfortunately, we don't have access to many HLICC patients, so we've had to resort to databases, and there is a very important data set generated in NIH, and this shows that indeed when we take HLICC samples, interferon response is highly upregulated. This has been now confirmed also in a variety of Chinese cohorts, where they find that indeed there is a strong inflammatory signature in the patients. We were able also to perform the convolution of back RNA sequencing from different kidney tumors from the TCGA. And what we could see is that the fumarate hydratase deficient renal cell cancers are characterized by a very striking inflammatory milieu, meaning that both lymphocytes and myeloid cells are much strongly accumulated in the microenvironment compared to, for instance, other kidney types, kidney cancer types. So this overall suggests that indeed, even in patients, there could be an inflammatory signature, and we are currently investigating that. Now, the other question that I want to maybe address with you now is whether, given these pro-inflammatory effects that we saw in epithelial kidney cells, we ask whether this signature is also conserved in immune cells. And here I am presenting some work that we have done in collaboration with Leuconeum in macrophages. And why we stumbled upon this, because when we perform metabolomics on LPS stimulated bone marrow derived macrophages, we found and we made this striking finding that along with itaconate that is notoriously highly accumulated in macrophages, we found also fumarate strikingly accumulated. At the time when we did this analysis, we almost ignored it, but eventually the more we investigated, the more interesting the situation became. We found that LPS gives rise to decrease in fumarate hydratase. This is accompanied by an increase in fumarate. And eventually, without going into many details, we found that in macrophages where we could inhibit fumarate hydratase with a phage inhibitor called fumarate N1, we could elicit a similar inflammatory response. You can see here they measured interferon beta, and this was dependent on the release of mitochondrial RNA. And here I put this last piece of data that might be also more relevant for you, is that we found that this connection between fumarate hydratase loss and inflammation seem also to be present in patients from lupus. This is a sample set where we found that fumarate hydratase is strikingly suppressed in patients with systematic lupus, and this could probably, at least in part, explain part of the inflammatory signature. This is eventually the summary of these two findings. In two different cell types, we found that fumarate hydratase loss can give rise to aberrant mitochondrial function release in our renal cancer model to mitochondria-derived vesicles and SIGA-STING activation. In macrophages, for reasons that are not clear, the mitochondrial RNA seems to be more important and more importantly linked to MAPS, but eventually and the convergence signature is the same. So with this, I would like to conclude, and I just want to thank the people in the lab, and I already mentioned all the people involved, and I would like to thank the collaborators, in particular Julian Prudent, who was instrumental for the work on mitochondrial DNA, and all the other collaborators. And thank you for the attention. I'm happy to answer any questions you might have. And thanks a lot.
Thank you kindly, Christian. That was quite a journey you brought us on from the beginning of biochemistry into, mechanisms, and now maybe some even associations with inflammatory diseases and all of that. Beautiful, beautiful work, and very, very nice presentation. While we wait for people to post their questions in the Q&A, maybe I'll start with some questions.
Shall I stop sharing or
whatever you prefer? That's okay to do.
Okay, maybe I will leave this schematic here just in case people want to discuss it.
That's great. And some of my questions you brought nicely up at the very end, and I think you and I can maybe discuss them in a little bit more detail later on. But I was surprised to see but happily surprised to see the reduced levels of fumarate hydratase in lupus. Because as you said, inflammation or TLR signals should bring it up, should increase these levels. Why do you think that they are reduced in lupus? Do you think it's a genetic issue, or do you think there are other kind of regulation of my question is it a cause or consequence of the disease, you think?
Yeah, this is going to be highly speculative. But
Yes.
we found that at least in macrophages, when they are activated by APS, fumarate hydratase goes down, and
Oh.
maybe, yeah,
Hmm. Hmm.
yes, and therefore maybe what happens in lupus, I have not much knowledge, and you're definitely more knowledgeable than me. But what could happen is there might be something that triggers this loss of fumarate hydratase and might contribute to inflammatory cascades that we observe in patients. Again, it's all correlative, but we found that in HLICC patients, there are uh, some families that have lupus. And in that case, for instance, the trigger might be genetic. In other example, maybe it's not the genetic, maybe it's environmental. But the more, yes, this is still speculation.
No, that's all good. And follow up on that, I suppose, is that our patients and many patients with autoimmune diseases have an increased prevalence of tumors. Do you think that these metabolic changes could be part of that,
Yeah,
or is it?
that's interesting. Um, for sure, if in these patients there are some intrinsic metabolic changes, these can, especially in the tissue microenvironment, that could, predispose or increases the chances of transformation. I don't know whether we can say it can trigger or drive, but it can certainly, push the, the threshold.
Wonderful. We have two questions. I don't know if you want to bring them up. Otherwise, I'm happy to read them
Yes,
out for
yeah.
you. Yep.
Oh, yes. Okay. So here I have a question from Thomas. Can you tell us a bit more about the SNX9 mechanism? Yes, I can. I can go. Yes. Or well, here. So the reason why we stumbled upon SNX9 was again serendipitous. When we look at how mitochondrial DNA was released, we performed a mini screen of players that at the time were known to regulate mitochondrial DNA release. And therefore, we knocked out VDAC, we knocked out BAX, we also regulated all the mitochondrial dynamic related proteins and XNN9, SNX9, because it was known to create this vesicular structures that carry mitochondrial DNA. And only when we knock down SNX9, that is an enzyme that pinches off membranes of the mitochondria, we could block the entire inflammatory cascade. So at the moment, we don't know how these vesicles are formed or what triggers them, but we know that the vesiculation of mitochondrial DNA depends on SNX9. And when we knock down SNX9, what we observe, quite intriguingly, is that these bubbles here are still formed, but they are not completely removed. So you will see the mitochondria with a lot of bubbly surface, but due to the absence of SNX9, they cannot be detached and completely removed from the mitochondria. I hope, Thomas, this is okay. Okay. But I think I have a meeting with Thomas later. Maybe we can discuss. Okay, here I have another question from Bobby. Is there any way to increase fumarate hydratase level? Any roles of diet on fumarate hydratase? Yes, good question. At the moment, there is no way to increase fumarate hydratase level, but probably one could increase fumarate hydratase fluxes, just changes again metabolic fluxes, but this is tricky and I don't think anyone has tried so far. And the same for the role of diet on fumarate hydratase, there is very little. Actually, it will be surprising that it is not even known what regulates fumarate hydratase transcription. Okay. I have another question. Yes. What sort of therapies do you envision for treating patients with this renal cell cancer, based on your study? Yes. So this is very important. As I alluded earlier, one approach that we are attempting now is to use this metabolic vulnerabilities to either prevent or to treat these tumors. What I mean is that these patients are known, and technically we don't know when the tumors appear, but we know what nutrients they need to survive and to proliferate. So the idea is to maybe deprive these patients from arginine or reduce cysteine, or maybe even give some hemoxygenase inhibitor. This is still, at the preclinical stage, but this likely blocks specific metabolic pathways that only occur in this fumarate hydratase deficient cells and needed for the survival. So this is one way that we are attempting. So it will be maybe adjuvant. So I doubt that this will work as a sole therapy, but we are trying combination as well with other standard therapies for renal cancers, such as TKI and so forth. Okay. Question. I don't see any more questions here.
I think those were all the questions for now, unless someone else sends a question. I think we'll sum up here, and I'll save my questions for our
Yes.
upcoming talk. But again, a wonderful discussion and wonderful presentation. Thank you so much for coming and sharing
Thank
of your
you.
insights with us. And I think that ends the session.
Thank you very much, and thanks everybody for listening to me. I appreciate it.
Thank you.